So now we are going to transition over to our research update on CureGRIN funded projects. So we have some exciting insights from Dr. Law and Trinella's team funded project, Dr. Frankel and Dr. Sands funded project, Dr. Ramsey, Dr. Kalev funded project, as well as Dr. Bowie and Dr. Coombs. So we'll get started with Dr. Law if you would like to share your screen.
Thank you so much. Okay, let's get this started. Okay, I assume you can see my screen?
If you want to swap displays, it's on the presenter view right now.
Okay.
Perfect.
Perfect. Okay. I will give a brief update on several projects because the one which is funded is still early stage. Took a little bit longer because I was moving last year from Cleveland Clinic to the University of Texas Houston. But nevertheless, we worked a lot on GRICON and as a whole. And I just want to, I try to be fast. Okay. So first I will talk about, just very briefly, just want to encourage everyone, just what just discussed, to participate in research because it really makes a difference. So, and particularly, we are moving more and more in the direction that we go away from this kind of framework where data is collected by people and then you can apply for it, which it basically removes all citizen scientists or away from this data where you don't have institutional support. And typically this also makes it challenging for people who cannot code to access this data and analyze it fast and easy. So what more and more people do is developing online resources where you can explore the data. And what Tim just presented is really that for the GRIN community, we really made this work. So we have now the GRIN portal and more data is coming. So there's still some data which we don't have from clinical trials, for example, and I hope this will change now. But in general, just participate in online in all these surveys because the data will sooner or later ends up that you yourself can actually explore your own data in comparison to others. So here's just briefly how we built it. And just want to show you that the GRIN portal was the very first we developed and still the most advanced ones. You can be really proud of this. So you have so much community support that we can really, build a lot of features into this, but I don't have time to discuss this. But this is now growing from GRIN to other portals. It's really nice. And this is how the GRIN portal looks today. And you can just explore this. Tim already gave a shout out, but I just really encourage everyone to go to this page and this is data you have entered, so we do something with this every day. And here really to make the point, right? So with our patient portals, we have now 150 visitors every day. 150 people exploring, the data you enter and with also the functional data and so on and so forth. And other than that, this would be all behind publications which no one can read and they're behind paywalls and it's super hard to get. But with online resources like the GRIN portal, people all over unfortunately not all over, we need to translate them, right? So there's more work to be done. But you see, this is, what people use this. People talk about rare disease, but globally rare disease is not rare, right? So you know this. So there's more to be done and more to explore, but in general, online resources, people use them. Just want to make the point. Okay, so and here's the team, for the GRIN portal. And I say this every time, this list can be way longer, just about all the people who are doing stuff. Okay, so let's move to the second part. I mentioned this before. So we, since we have now all the data from the GRIN portal, we saw patterns, right? So here's the NMDA receptor. So the protein which is encoded by GRIN1, GRIN2A, GRIN2B, GRIN2C, GRIN2D. And you can see, for example, that the patient variants are located on the structure, very different compared to variants which are seen in people without disease. So every gene and every protein has mutations, but some of them can cause disease. And you can see them clearly here that they are different positions just by eye, right? So I always compare this to a car. So imagine this would be a car and you would see specific positions when you have like a dent, then the car would not drive anymore. For example, if you have a dent or a hole in the tires, then you have a problem. But if you have a dent somewhere on, I don't know, somewhere in the back of the car, then like here, then you don't have a problem. Okay, so it's just the statics which change. So this is how we approached this, and we saw this already like 2 years ago. And we did all kinds of analysis and learned that it's related to some of the biology associated with that channel. And I don't want to go much into detail. But the point is, we see very different patterns even within the patient variants in terms of what they do for function. So for example, the variants which lead to less functioning of the channel are located at a different position compared to those which increase the function. So, and this is really cool. If you can see this already by eye, right? So you can develop your own AI algorithm just by in your head and see, if the variant would, if we have a new patient variant, it would be located here. Potentially, it's more likely to cause loss of function compared to a new patient variant, which would be here. You would get just, intuitively think might be more gain of function. So in research, we can formalize this and we can do some algorithms, and this is what we did. And so basically, we can develop an algorithm which predicts pathogenicity better than everything else at the moment. And we can look at VUSs and can reclassify them into benign or pathogenic with certain level of accuracy, but I don't want to go too much into detail about this now. And we can also develop a function prediction score. So, and this is now finally final. It took a long time to refine this. And there's a lot of work with Steve Tarnellis group. So to get really the variance right, it's, there's so much consideration to call something a loss of function or a gain of function, and so much to consider when you then develop algorithms on this. So we had already similar results 2 years ago, but we consistently, and particularly Ludovica from my team, worked on this to get it right now. And so we finally have it, and we integrated this even in the portal for research purposes. It's below in the bottom of the variant analysis fraction. So you can enter any possible variant, literally any possible variant for GRIN to A, GRIN to B, GRIN to C, GRIN to D, and GRIN 1, and it gives you this prediction based on the data. So that's cool, right? So the more functional data we have, this can be more accurate, and you can be certain that you cannot functionally test everything. So having algorithms increases the broad application of variant function interpretation. So this is not still work in progress, but we're moving here. And finally, also since we are finally done with this, there is a paper now which we will submit, or Ludovica, who's on the call, so she's not under pressure, will submit in the next days, early next week latest, the publication to the journal, and at the same time we put it in the medical archive as preprint, so everyone can read it hopefully by Wednesday. So it should be online and you can read it and criticize us. Okay, so the funded project, so again, we're doing a lot of GRIN work, unfunded. So it's using these algorithms or these predictions which we have developed for the GRIN genes and trying to transfer it to GRIA. So the hypothesis is, if you, for example, if the GRIN gene would be a Ford sedan, and the GRIA gene would look similar to, let's say, a Toyota sedan, then maybe we can, what we know what is bad for a sedan, which from a one brand might be bad for a sedan or for another brand. So for example, if you have like this dent in somewhere in the back of the car, might not be bad, but if you have a hole in the tires, might be bad. You might learn this by studying the Ford sedan. And then if you see a Toyota sedan, you might think, hey, maybe the rules are here the same. And this is basically what we try to do with these different genes. So here is the latest number of variants which we included from the GRIA genes. And that's also the rationale why we have to do this. For the GRINs, we have way more variants and we have a tremendous amount of functional data. So this allows us to develop algorithms. But if we would just do this with GRIA, we couldn't do this because we have two less variants or patient data and functional data to develop algorithms just for GRIA yet. So in the future, we could do it just with GRIA, but at the moment we don't. So that's why we want to take all the knowledge and resources from the GRIN genes and try to transfer it to the GRIAs. So, and we are very hopeful about this. And you can see just by eye that there's specific patterns where you have the patient variants versus the benign variants. So, for example, the patient variants cluster in specific regions. And there are also some regions where all the benign variants are. But it becomes more extreme even if you look at variants from the general population. So like almost 1 million people who don't have developmental disorders. You can see here in gray where a lot of these variants are from people without developmental disorders, 1 million. And you can see there's some holes, right? So you see here, okay, it's pretty obvious that here you become a patient, right? So here as well. So, So, and we can use this information. So very hopeful, because this is some proof of concept which we needed to see. Once we see this, good. And so that we can develop now, again, this algorithm approach where you take one data set which has a lot of data and try to transfer it into ones where you have less. So in this case would be the GRIN genes, and you would try to transfer this information and the algorithm slightly and modify it that it works for the GRIAs. And this has been shown, so we didn't come up with this, this has been shown in other disease domains. So, and yeah, here's other examples where people do this. Okay, so why do we believe that will work? Because if you look to the left, this is the NMDA receptor, which is based on GRIN1, GRIN2A, GRIN2B, GRIN2C, GRIN2D, depending on which configuration. So the GRIN genes, and here's the AMPA receptor, just based on the GRIA genes. So, and you can just see by eye, they're organized very similar, right? So if you would be a biologist and would have seen like thousands of, receptors or genes, you would see this is unusually similar. This basically, if you look at across humans, you would say these are twins. They must be related. And because they are. So basically on evolution, they are related. And since they look very, very similar, we think also that the mutations do similar things. It's the same thing. If you look at thousands of cars, if you look at two sedans, which you would expect that two sedans have similar properties compared to another car compared to a motorcycle, right? So you would think something which looks similar works similar. And this is the underlying hypothesis, and it's very strong here. So what Ludovica from our team did now is that she actually confirmed this with a little bit more bioinformatic analysis, and it looks really strong. We're very happy about this. Don't want to go too much into detail, but you can always reach out. And then we do even more analysis where we try to see where we actually are. So because we know that analysis we did within the GRINs, so like for example, GRIN2A, GRIN2B, we needed to get this level of scoring, which showed that it works perfectly. So that's what we learned in the past. For the GRIN2A versus GRIN2B, this works perfectly. They almost work identical if they have this kind of scoring. And if we look across the GRIAs, we know that they are even more similar. So GRIA 2, 3, and 4, GRIA 1, 2, 3 to 4, they are more similar to another than GRIN2A than GRIN2B is, which is perfect. So even if you don't want to do transfer learning from the GRINs to the GRIAs, just take in every basically functional readout, or functional readouts between GRIN2A, GRIA 2, and GRIA 3 and 4 should be very similar. So you could actually take this information within this group quite certain. So, and we can also see that we have a very good relationship from GRIA 1, 2, 3, and 4 to GRIN1. So they map structurally very similar to this. This is what we see here. You can see very clearly by eye that the bluish versus the red, protein parts are similar if you align them. So you could even, if you push this over, you see that they even would really nicely fit. So this gives us a lot of confidence that we can do the transfer learning. So more confidence than we even had before. So really strong. And here's a different version of doing the similarity metrics, and it's even more confirmed. So there's more bioinformatics here. Okay, remember when I showed you the GRIN genes on the NMDA receptor? So GRIN is making the NMDA receptor, showing here that we see this pattern of patient variance versus population variance. So here, positions where mutations cause disease, positions where they don't. The nice thing is if we take the GRIA variants and put them onto the GRIN, GRIN1 position in the NMDA receptor. So basically, if we take GRIA variants and put them on the NMDA receptor, we see the same patterns. So like it's nicely here. Here's all the patient variants. Here's our population variants. So we see nicely here the population variants and benign variants. And we can see more variance in this area. It's like we see here. So this gives us a lot of confidence so that algorithms will work. And so here, more specifically, if we split it by gene, so here the GRIA variants on GRIN1, GRIA 2, here's GRIA 3, and it's nicely here where we want them to have. So that's why we are we're very certain that we can even take the GRIN prediction score and use them for GRIA variants if we transfer the GRIA variants on the structure. So really promising results. But there's more work to be done. The project was just initiated. Because of my move, it took a little bit longer. Sorry for that. But Ludovica is really working on this now, and we are very all the work which just looked, what we just saw took actually quite some energy to get there, but we have it now, and we're very optimistic that we can use the GRIA the GRIN information to inform the GRIA work. And we're collaborating with Steve Charnelis to generate more GRIA functional data, and already in touch with Alan and the GRIA clinical and wider functional community to also get data from these teams, and then put everything into a GRIA portal. And here, most of the work which was presented today was done by Ludovica, and with this I stop. Thank you very much. And of course, thank you, CureGRIN, allowing us to work on this.
Thank you so much, Dr. Law. If anybody has questions, you can put them in the chat for now. Hopefully we'll have time at the end of this session to be able to ask those. And then we're also going to be transitioning to our Ask Me Anything anyway, so we'll be able to capture some of those questions if they get missed. So now we're going to move on to Dr. Tristan Sands from Columbia University, who's collaborating with Dr. Wayne Frankel on a CureGRIN funded project. Dr. Sands, if you would like to share your screen. I can see your screen.
How's that?
Perfect.
Okay, great. So I wear two hats. I'm a clinician at Columbia, and I see I'm part of the same group as Jen Bain, who's on this call. My focus is treating epilepsy, but I'm also a translational researcher, and I work in collaboration with Dr. Wayne Frankel using mouse models to develop and test new therapeutics for patients. And I just want to thank CureGRIN for the opportunity to present this work, which was funded to investigate a therapeutic strategy for GRIN2A in collaboration with Dr. Frankel as well as Dr. Harper's lab at Nationwide Children's Hospital. So in 2020, Dr. Frankel and colleagues on this call reported investigations into a variant associated with DEE, or developmental and epileptic encephalopathy, the S644G variant in GRIN2A. And it was demonstrated to be gain of function in that it increased the open probability of the channel and slowed the deactivation kinetics. The variant occurs in a highly conserved sequence. It was de novo in the patient. And the group reported a mouse model where the variant was introduced through a knock-in approach. And the heterozygous mice show an array of phenotypes, including altered seizure thresholds and differences in their performance on neurobehavioral batteries, as well as some subtle differences in hippocampal morphology. The homozygous mice, so the mice that have two of these disease copies of GRIN2A, actually had their survival limited by lethal seizures. And the two phenotypes that were the focus of this study in terms of developing a rescue therapy were the lethal spontaneous seizures that led to early demise in the homozygous mice carrying two copies of the disease variant, as well as altered seizure threshold to electroshock with the 6 Hertz limbic seizure electroconvulsive threshold paradigm. And the heterozygous mice actually demonstrated a protective effect. So they were actually harder to provoke seizures in compared to wild type. And so the approach that we took and was sort of stated earlier by Dr. Frankel in his talk, that one approach to gain of function disease would be to reduce the toxic gene product through a knockdown approach. And as he mentioned earlier, this kind of approach has as one of its as pluses that it's agnostic to the particular variant and might be effective for other gain of function variants as well. And so collaborators at Nationwide Children's Hospital and Scott Harper's lab screened through a number of constructs seeking to knock down GRIN2A in a luciferase assay that construct, one of the construct was selected to clone into AAV9. And then we did experiments injecting intracerebroventricularly at P1. A lot of this work was done by a talented scientist in the laboratory, Divya. And the idea, again, behind this approach was to see whether or not we could mitigate the severe phenotype through this sort of non-selective targeting of GRIN2A. The idea being that the benefits of reducing the gain of function may outweigh the impact of creating a partial loss of function. And so these are the results with the homozygous mice, which, normally are dying by the end of the first month or so. With the injections of the RNAi vector, we can actually extend the lifespan out almost 3 times as long, depending on the dose of the injection. And we used a number of different doses, but there seems to be some sort of limitation in that going higher didn't necessarily buy us any improved survival, as you can see from the graph. And then just to sort of contextualize this, with respect to the repurposed drugs that were reported in the 2020 paper, looking at effects on the survival phenotype, those treatments really were sort of akin to what we saw with the low dose, and so we've sort of, they're sort of limited to this area here. So this is a promising sort of approach to tackling the gain of function variant. And quickly, just to show you the heterozygous mouse phenotype, the threshold to the 6 Hertz limbic electroshock, we see a partial rescue in that the treated mutants move a little bit closer to the wild type animals in terms of their seizure threshold. So again, sort of arguing that we're having an impact on the severity of the condition and reducing it to some extent. There are interesting things that we see in the surviving mice that were unexpected. So again, homozygous animals don't usually survive beyond the first month. If you look at the way wild type animals walk, that's the way a normal mouse is walking. These survivors had a funny sort of gait that we've been calling sticky feet, that Dr. Frankel came up with a term, sticky feet. And it kind of captures something, because the mice sort of walk in an awkward fashion, and it's not really clear. We don't see this in wild type animals that we treat with the treatment, or heterozygous animals, just the surviving homozygous animals. And so the thought is that perhaps this is a sort of emergent phenotype because the animals are surviving, whereas you normally wouldn't see it. And so the conclusions are that this sort of non-selective GRIN2A knockdown approach with the gain of function variant was capable of shifting seizure phenotypes in this mouse model towards the wild type, and so making the condition less severe. But higher doses don't necessarily improve survival in the homozygous animals. And I didn't show this data, but higher doses were also associated with some deaths in heterozygous animals and even wild type animals. And so there are limits to what this kind of approach can accomplish on its own. And then the final thing to say is that there's this unusual sort of motor phenotype emerging in surviving homozygous animals that we haven't appreciated up until now. And so I just want to thank everybody in my lab, especially Divya, who along with Dr. Frankel and I did the work, and our collaborators at Nationwide Children's Hospital, and to my patients and families. The families of my patients are my heroes, and they motivate what I do in the clinic and in the laboratory. And I just want to thank Wayne for including me in this collaboration, and for CureGRIN for the opportunity to investigate this treatment strategy.
Thank you so much, Dr. Sands and Dr. Frankel. Next, we're going to transition to Dr. Maggie Kalev's presentation. She is at the University of Auckland and is collaborating with Dr. Amy Ramsey at the University of Toronto. So Jeff, if you want to key that up.
Hello, my name is Maggie Kalev. I'm a hematologist scientist at the University of Auckland, working with Dr. Amy Ramsey and her student Sylvia Okafor on a project to uncover how we can use blood cells as the source of potential biomarkers for GRIN disorders. The rationale for this project is that essentially all blood cell types have now been shown to express NMDA receptor components, although receptor functionality still needs to be better defined in these cells. In Toronto, Dr. Ramsey's student examines peripheral blood changes in 2 transgenic mouse models of GRIN1 variants. Sylvia will present some of her data in her part of the report. In Auckland, we support Sylvia with this analysis. She sends us digital scans of bone marrow and peripheral blood from mice, and we examine potential shifts in progenitor populations and cell appearances. But most importantly, we want to validate that changes detected in mice can also occur in human cells and in patients. We focus on platelets because we have most expertise in this area. We also focus on plasma because plasma can be easily obtained and may carry substances released not only from blood cells, but also from the brain. We would love to analyze complete blood count results from patients. We prepared a list of relevant blood parameters and a brief questionnaire to help us interpret these results. Multiple acute and chronic conditions can impact blood counts, and these include seizures, infection, heart, lung, kidney, liver, bowel, blood diseases. Therefore, it is important to understand the context at the time of testing, why a particular test was done, drugs patients are taking, or if there are any particular nutritional factors contributing. There are obvious logistical barriers as to why such data cannot be obtained quickly from a lot of patients scattered across the world, but we will continue interacting with the GRIN registry to determine what is possible and help where useful. We also work with Combined Brain to obtain blood samples from GRIN patients for molecular studies. And this is a list of samples currently available. There is samples from 16 patients, 5 from GRIN2, from GRIN1, 3 from GRIN2A, 7 from GRIN2B, and 1 from GRIN2D. We want to test these samples together with the plasma from mouse models to look for abnormally expressed molecules that could serve as potential biomarkers. Unfortunately, this current set of samples does not include any cellular material, and there are no controls from healthy age-matched children. Current controls are mostly from parents, so this will need to be addressed. We want to ship these samples to a center with established expertise in mass spectrometry work for biomarker detection, and we are currently reviewing our options for this. In the meantime, to assist in examining the applicability of mouse findings to patients, we are generating, using CRISPR-based editing, genetic models of GRIN1 variants in human MEGA1 cells that have both platelet and red cell differentiation potential. We have chosen these 5 patient variants, 2 in green, a loss of function, and 3 gain of function. in red, and one of these variants, the Q536R, aligns with Dr. Ramsey's mouse model. We acquired a set of base editing plasmids. For most, the Cas9 enzyme has been cloned together with the green fluorescent protein. We designed guide sequences for each of the chosen variants and cloned them into guide RNA plasmids. MEGA1 cells were successfully transfected with GFP-expressing cells using liposuction. These are GFP-positive cells from the whole pool of transfected cells. The efficiency is about 10%, which is a little bit low. Therefore, we are working to increase this efficiency using nucleofaction before isolating single positive cells to generate clonal lines. In parallel, we made steps to import a set of induced pluripotent stem cell lines derived from GRIN patients that were generated by Dr. Zhajing Wen in Atlanta. These lines are currently used for neuronal work, so we would be the first ones to apply them to blood studies. We have chosen these 3 GRIN1 lines for this, 2 loss of function, including the Q536R as in previous studies, and 1 gain of function line. We want to differentiate these IPCs and genetically modified MEGA1 cells into blood cells to determine how GRIN1 variants impact platelet and red cell differentiation and function. We also want to identify molecular changes in gene and protein expression, particularly for molecules involved in inflammatory and cell stress responses and calcium homeostasis. This focus is informed by the exact type of changes that we have already found in our GRIN1 loss of function MEGA1 cells, and that Sylvia found in her murine models of GRIN1 variants. Please email me with any comments or suggestions. For example, CBC monitoring and sample collection may be a worthwhile addition to some of the clinical studies that are being conducted. Or CBC testing may already be done within some studies. If so, it would be great to use such data for analysis. I'm happy to share the list of parameters and the questionnaire or sample preparation protocols to help standardize how the data and samples are collected from patients in different parts of the world. Thank you for listening, and I should be online to answer questions. Bye.
We're very grateful for Dr. Kalev's presentation. We also are going to hear another part from Sylvia Okafor and Dr. Ramsey. Jeff, if you want to key up that video.
Hello everyone, my name is Sylvia Okafor and I'm a PhD candidate in Dr. Amy Ramsey's lab at the University of Toronto. And today I'll be sharing some of my findings in relation to analyzing hematologic NMD receptors in mouse models of GRIN disorder. Of our study is to examine the effects of GRIN disorder on blood cell processes with the aim of revealing potential biomarkers of disease that are hopefully clinically relevant. So far, we've discovered several blood biomarkers for GRIN1 disorder using these mouse models here. The first is the GRIN1 Y647S, which is a gain of function patient variant, and the second is the GRIN1 Q536R, which is a loss of function variant. And after completing a complete blood cell count, we found evidence of increased red blood cell proliferation in the GRIN1 Y647S, with that entailing increased red blood cell count, increased hemoglobin, and hematocrit, although these don't seem to be clinically significant. And the second finding was enlarged red blood cells and platelets for the GRIN1 Q536R. After observing these changes, we then wanted to see how calcium flux was changed within red blood cells, since we were seeing some obvious changes within the blood cell properties. And so we then did calcium imaging with red blood cells collected from the Y647S and Q536R mice. And our findings revealed that upon the application of NMDA, just realized my pointer is not on. Upon the application of NMDA on the Y647S red blood cells, we found that there was a significant increase in calcium flux after the addition of NMDA. And in contrast, the Q536R, after the application of NMDA, there was no real response to the agonist, which seems to indicate the presence of gain of function in the GRIN1 Y647S and loss of function in the Q536R, which is in line with what the CFIRV database has published. Since we saw these changes, we then wanted to examine the JAK2 STAT3 pathway, which is involved in many processes of cell proliferation and differentiation, as well as in immune function. And there's evidence to suggest that NMDA receptor activity can phosphorylate, or in other words, activate JAK2. And so to look at this pathway, I collected bone marrow cells from the mice and processed them for flow cytometry to look at the phosphorylation of JAK2 and STAT3. And today I'm just going to be sharing some preliminary data from untreated bone marrow cells. And interestingly, there was a significant increase in phosphorylated JAK2 in the GRIN1 Y647S, and there was no other change present without treatment. And there was also no significant change in basal phosphorylated JAK2 or phosphorylated STAT3. And this here is just showing the percent of single cells that have either only phospho-JAK2 or only phospho-JAK2 and phospho-STAT3, or cells with only phospho-STAT3. So then, after observing these interesting changes, we then wanted to look at the white blood cell populations, and we again did some flow, but this time we used spleen cells. And the reason why we used the spleen is because they're very hardy, so there's a lot less of them cells, a lot less of the cells die, and they're easy to collect. And so we then looked at the splenic white blood cell populations, and interestingly, we only found atypical white blood cell count for only the GRIN1 Q536R, which was quite interesting despite our previous findings in the JAK2 STAT3 phosphorylation assay. Here on the top row, these graphs are just showing the percent of these cell types within the the spleen cell population, and the bottom graph here is showing the percent of these cells expressing GRIN1. And interestingly, there is a significant variation in the amount of GRIN1 present in these different cell types. And what was quite intriguing was that the CD8 effector memory T cells actually have approximately 50% of them express GRIN1, and there's a slight trend towards there being less in the Q536R. Although there was no change in the baseline JAK2 STAT3 signaling for the GRICON Q536R, it's clear that they are experiencing some sort of atypical immune cell communication, which perhaps could be leading to these changes in white blood cell population. So in summary, the calcium imaging data strongly indicates the presence of gain of function in the GRIN1 Y647S and loss of function in the GRIN1 Q536R, which is in line with the findings presented in the CFIR database. NMD receptor activity can regulate JAK2 STAT3 signaling and its downstream processes, which include cell proliferation, differentiation, and immune cell function. And these were present in the Y647S as increased red blood cell proliferation. And in the GRIN1 Q536R, this was presented through abnormal white blood cell counts. So in summation, we found potential blood biomarkers for gain and loss of function in GRIN1 disorder. And they're likely not pathological, but they will help point towards potential pathways we could explore to find more robust biomarkers. So I wanted to thank all those who helped me work on this project. And of course, I would like to thank CureGRIN for supporting our research. And yes, thank you for supporting us.
We're so grateful for Sylvia's presentation, as well as Dr. Ramsey's work and Dr. Kalev's work. Now we are going to transition to Dr. Bowie's presentation. Jeff, if you could key that up.
Okay, good afternoon, everybody. I guess the first thing is thanks to the organizing committee for CureGRIN for giving me this opportunity to be able to present to you today. If you're watching this video, then what that means is that I am still in the US trying to find my way back across by flight into Canada, and I've somehow got delayed and not been able to make it to this session so that I can actually present to you in person. So maybe I can just start by thanking CureGRIN for this opportunity, of course, but also because they've given my lab some funds, some seed funds to be able to actually look at GRICON disorders, particularly interested in a hypofunction in NMD receptors, so kids who are primarily affected by that. And the reason we're particularly interested is because we think we may have a therapeutic approach that may be useful in the treatment of kids with that kind of condition. So to give you a little bit of a background to why we're interested in this, my interest in the GRICON disorders actually comes from, maybe comes from a kind of funny place. It comes from the work that we've been doing on Fragile X syndrome, which we've accumulated quite a lot of work on this. Actually, we haven't yet published it, and there's a couple of reasons for that, which I'll get to in a little bit. But what was interesting when we looked at Fragile X syndrome, so in this slide here, and hopefully you can see my mouse, maybe I'll just convert that into a little laser pointer so you can see it. So here's a little kid who's got Fragile X syndrome. But what was very interesting about it is when you look at the features of kids with Fragile X syndrome, they've got cognitive impairment, they've got learning disabilities, they've also got social anxiety, impulsivity, aggression. There's also about half of the males have autism, and of course the occurrence of epilepsy, and I think epilepsy is actually higher in GRICON disorders. But in looking at that and the fact that I actually teach medical students, it really struck me that Fragile X and GRICON disorders actually have a lot of overlapping features. And because there are a lot of overlapping features, then we thought that, well, maybe some similar kind of aspects at the molecular level are going on in Fragile X and also in some forms of GRICON disorders. And so we found in the Fragile X brain is that we've actually uncovered a kind of, if you like, a novel signaling pathway that's mediated by NMD receptors. Now, I appreciate that much of the audience here may not be clinicians or research scientists, so I'm going to try and keep this mainly to kind of cartoon format. But the way we tend to think about NMD receptors, so here's our NMD receptor that you would find in the brain. It gets activated by glutamate. And then there's the transport of calcium through the NMD receptor. And calcium is a very important molecule that's been transported because it then signals inside the cell to a number of signaling molecules. And a very famous one is CAM kinase 2, which you should see here. And once CAM kinase 2 gets activated, then it leads to a whole bunch of different kind of biochemical events, which doesn't make sense to go into it in detail, at least in this talk. But it leads to learning and memory events. And that's how we tend to think about the molecular basis of learning and memory. And so you can imagine if you get a dysfunction in NMD receptor function, then that could actually have an impact on learning and memory capabilities of someone. But what we found is that in a healthy person as well, and healthy mouse models that we've been looking at, then the NMD receptor also does something else. So the NMD receptors also signal through another kind of enzyme called neuronal nitric oxide synthase. Neuronal nitric oxide synthase is kind of an interesting molecule because it does two things. It signals inside the cell and generates cyclic GMP, which is another signaling molecule that actually lasts quite a long time. It's inside cells, triggers a whole bunch of different kinds of phosphorylation events for the researchers out there listening to this. But the other thing that's interesting is it actually causes the release of a gas called nitric oxide. And I'll show you in a minute that nitric oxide actually can activate local capillaries, which are next to cells, which express NMD receptors and neural nitric oxide synthase, and cause a vasodilation. That's actually really an important thing because it causes an increase in local blood flow into the brain. So what we found in the Fragile X brain is that this is actually a very, very important signaling pathway. And in fact, if you go into our publications, you'll see that we've actually tied this NMD receptor into a whole bunch of different kinds of signaling events. And if anybody's interested, the scientists and clinicians in the audience, you can go and dig up our papers where you can read about how we've done this work. And so we show that there's a kind of homeostatic mechanism. So there's a mechanism that both increases the excitability of neuronal circuits, but at the same time brings down the excitability of those circuits. And at the same time, concomitant with all of that, you actually get a local increase in blood flow. So you might measure that with fMRI, for example, in an intact brain, where you get a local increase in blood flow, you get this vasodilation, which occurs. Now, the reason I'm telling you all of that is what we found in a mouse model of Fragile X syndrome. We found that there's much, much fewer NMD receptors in certain types of cells which express neural nitric oxide synthase. And we found this in different kind of brain regions. And it looks like we may have also found it in adult tissue as well. And what that means is all these learning events, all these biochemical events that lead to learning within individual cells, including the vasodilation, are actually lost in the Fragile X brain. And that's really something quite significant, and it's something that no one's really appreciated before. Now, the link to the GRICON disorders is because the observation is really tied into a reduction into the NMD receptor response. All the other signaling pathways are actually intact in the Fragile X brain. Then what we think is we look at a hypofunction of NMD receptors in a mouse model, for example. If you look at that one, we think that all of these same signaling events will be deficient, including the failure to actually induce vasodilation. So I'm not going to take you through all these different kind of experiments we've done. So I'm going to show you something that's much more visual for everybody. And that's by looking at, so if you just give me a second, I'm going to actually switch this for a second back to this. So what I'm going to show you is an experiment where we've actually looked at local blood flow in the brain. And on the right-hand side here, you can see a normal wild-type mouse capillary. It's actually a capillary here. And if you look very carefully, there's a little cell here called a pericyte. You'll see in a minute that the pericyte actually can cause a contraction, but is a dilation as well of the local capillary. And here's exactly the same experiment. You can see that we've done in a Fragile X capillary, exactly the same experiment. So what we do in these experiments, we actually induce a local vasoconstriction of the capillary. You'll see that. And then when we activate, and this is a normal mouse, so we activate the mouse's NMD receptors, you'll get a vasodilation. So you'll see that in a minute. I'll just indicate when it happens. So look carefully here. You can see there's a vasoconstriction. And then when you activate the NMD receptors, you can see you get this vasodilation occurring. These little donut-shaped things here are actually your red blood cells. You see the vasoconstriction happening again. So I'm going to play that again for you just in case you missed it. So here's the vasoconstriction that occurs, but then when you get an activation of these NMD receptors, you then get a vasodilation. You see that occurring, and that would cause a local increase in blood flow into the brain. And that's a very critical thing, because when you're trying to learn something, then it's very important that you get oxygen and all the nutrients that is needed to drive and maintain the pathways within the nerve cells that actually drive that. However, when you do exactly the same thing in the Fragile X, so it's exactly the same experiment, but now done in a mouse model of Fragile X, you can see that when we try to cause vasoconstriction, first of all, we don't get that. You can see the red blood cells, the donut shapes can bind. NMD receptor activation, because there's not much NMD receptors, you see you don't get the vasodilation as well. Now, what's compelling about all of that is that in the context of Fragile X, we've actually found a way in which to recover all of these vascular deficits and also the neuronal deficits within Fragile X. I'll tell you a little bit more about the how complicated this whole thing is within the context of the local capillaries. So what we're seeing here is that from the nerve cells, the NMD receptors are releasing nitric oxide, and there's a number of different things going on. I won't take you into details, but here's a little parasite showing here. And when you get activation of these parasites, then you can cause vasodilation, and that causes a local increase of blood flow into the tissue. And we think that's a really important thing that needs to occur for learning to occur. In addition to the deficits occur in terms of the neural signaling, you also get deficits in the local blood circulation in these cells. So you might say, well, why is that important? So that's just one cell in a cell full of billions of different types of neurons. So the reason we think that's important in the context of the work we do in Fragile X, and also in the context of how we think it may play into GRICON disorders, is because cells which express neuronal nitric oxide synthase. So here's the original observation from David Brett and Saul Snyder, who are working at Johns Hopkins, where they cloned neuronal nitric oxide synthase and looks at expression. You can see it's very high in the cerebellum. This is a rodent brain, high in olfactory bulb. You can't see it very carefully, but it's actually found throughout the cortex and the hippocampus. So these are the areas of the brain which are incredibly important in learning and memory. But it's only seems to be found in certain types of interneurons, and that's important because it looks like many of the neurodevelopmental disorders, including GRICON disorders, seem to be disorders of interneurons. And so that's why we think this might be quite significant. Okay, so it's found throughout the central nervous system. Now, the other thing that was a bit of a clincher for us in terms of thinking about it in the context of Fragile X, where there's been more behavioral work done, is when we started to look at the behavior. We've done some of this, and we've looked at other people's work. Just one second. So when we look at the behavioral, so here's a bunch of different kind of behavioral tests, which I'm not going to take you through in great detail. But when we take these behavioral tests that you find in the Fragile X mice, so these are mice which have Fragile X, and then you look at patients which have Fragile X, then what you see is quite a good concordance between the occurrence of a reduction, for example, in pre-pulse inhibition. There's an increase in hyperactivity. The other thing that's very, very interesting, particularly because it occurs only in the males, there's an increase in aggressivity. So there's an increase in aggression in both the Fragile X mice, and that all seems to be found in the boys with Fragile X. And of course, that's something that people have noted with some of the kids who have got GRICON disorders. But the other thing is when you get a reduction in neuronal nitric oxide synthase within mice, so this is another set of mice that can't really generate nitric oxide, then what you can see is you get a very nice match between the behaviors that you see in Fragile X in the patient and also in the mice, but also in mice which do not have, are incapable of producing nitric oxide, the signaling molecule. We think we're going to get exactly the same kind of thing when we look at a mouse model, a hypofunction of NMD receptors in these mouse models of GRICON disorders. So just to finish in the last few minutes, I just want to tell you about where this is going. So when I started to present this within the university, McGill University, they realized that this was something that if we wanted to actually make it impactful for patients, then it was important for it to be translated. So out of that, we built this spin-off company that has gone for the last few years. It's called NOS Pharma. I'm sort of involved in directing the science side of things, but they've got two teams, a CEO, and we've recently recruited in the last year, so David Baker who's got more than 30 years in the pharma industry. He's also got a long-held interest in neurodevelopmental disorders and trying to bring drugs like that to the market. So that's another important thing. So what we're hoping to be able to do is to get proof of principle within these mouse models. And then what these two guys are going to try to do, and they're doing it right now, is to try to raise funds so that we can actually try to do clinical trials. And if anybody goes on to the website from NOS Pharma, you'll see that the CEO and the team there have actually acknowledged that my lab's been given some CureGRIN. So they're talking about how NOS Pharma has joined forces with CureGRIN to try and do that. So I think I was only asked to give about 5 or 10 minutes of a presentation. So that's the work that we're planning to do. We just got the funds from CureGRIN the last few weeks. So we're just basically getting everything set up. And so in the next few months, hopefully we're going to be able to do some experiments. And we're teaming up some other people. I'll tell you about that a little bit later. I'm actually in the US for part of that reason. We're going to be teaming up with another lab here who's got a real great expertise in NMD receptors. And so hopefully in the next few months, we'll be able to get our first data set, and I can maybe get back to you and tell you how we're getting on. So just to finish up, I just want to thank everybody in my lab here, some of the people who are involved in it. We've actually got new people in the lab as well now. I want to thank our funding that's kind of got us to this point. And of course, I want to thank the funding from CureGRIN that's really made this possible. So thanks again. And hopefully I can make it to the make it so I can give this presentation live. But if not, for the moment at least, they're going to have to unfortunately listen to me by this video. So anyway, have a good afternoon, and I look forward to meeting everybody the next time the community of CureGRIN get together. Thanks again.
Wonderful. Thank you so much for the update, Dr. Bowie. Okay, next we are going to transition to Dr. Ian Coombs. And I think he's here live if he wants to share his screen.
Down there we go. Can you hear me okay?
Yes.
Oh, that's brilliant. Okay. Well, thanks very much for the invitation. And of course, thanks very much to CureGRIN for agreeing to fund our project. So what I'm going to talk about today is just a kind of a necessary first step we needed to carry out before we could really start our work. And that is to try and understand the basis of different kinds of loss of function variants of the GRIA receptor. I was interested to hear from Dr. Lal earlier, and certainly this data that we're gathering here will eventually find its way into his system, either to test his modeling or to help build it, or both, of course. Where am I? There we go. So the title of our proposal was AMP receptor PAMs as potential precision therapies for GRIA loss of function. And face out the way. So there are a few terms here that you may well be familiar with by this stage in the day. I think I'm the very last presenter. So well done on your fortitude and your stamina to make it this far. So in terms of what these terms are in the title of our proposal, we have precision therapies. Now, these are drugs that work by directly counteracting the effect of a variant. So in terms of genetic disorders, if you can understand the basis of a genetic disorder, then potentially you can use the precise drug that helps to reverse the effect of that genetic change. And in terms of GRIA variants, we can, or in terms of receptors in general, we tend to think of them as either being loss of function variants or gain of function variants. So these are variants that either produce too little electrical signal in case of AMP receptors, or if they're gain of function, they produce too much electrical signal. And we're specifically interested in GRIA loss of function. And the particular drugs that would act as precision therapies in this case are PAMs. So this stands for positive allosteric modulators. These are drugs that increase AMPA receptor responses. And so putting all this together, you can see that if we can find GRIA disease-associated variants that produce a loss of function, then potential precision therapy to help directly counteract that loss of function would be a drug that increased AMPA receptor responses. And that's what we're hoping to look at over the next 18 months. Okay, but in terms of what I'm going to talk about today, this is work that has been going on for longer. The CureGRIN funding started in February. This work has been going on in the lab for quite a while now, and we've already gathered an awful lot of information that's going to be very, very useful as we move this project forward. And that's what I'm going to talk about today. Okay. So in terms of how glutamate receptors work, well, we know an awful lot about the shape of AMPA receptors, how the different bits of the receptor all come together. And get myself a pointer. And so they come together, the AMPA receptor subunits, there are 4 of them. They make what we call a tetramer, so four protein complex, labeled green, blue, red, and gold. And this particular receptor is pictured with an auxiliary subunit around the membrane region. So this is where the membrane is. And in grey here, these are called TARPs. These are auxiliary subunits that are very important for making sure AMPA receptors get to the synapse, and they modify their properties as well. And it's this combination of receptors that we tend to do our experiments on, AMPA receptors associated with these TARPs, auxiliary proteins. So now, while it's very, very useful to think about and to have knowledge of this molecular level detail of the receptors, so we know where all the different amino acids are, in terms of thinking about them as a little molecular machine, little moving entity, it's far easier to consider them as just these lumps. And I tend to think of them like a couple of Pac-Mans tied to a couple of sausages. And that's kind of how I'm going to depict the mechanisms of how they work. So, so these Pac-Mans are what we call the ligand binding domains, probably more kind of slightly stylishly called clamshells by the majority of people in the field. And they, each individual subunit has its own ligand binding domain, and each one of those can bind a glutamate molecule, shown here in green. Now, what happens when that glutamate molecule binds? Never shown this on Zoom before. I do hope this isn't glitchy, but when the glutamate binds in the ligand binding domains, the pairs of ligand binding domains are stuck together at the top here. And when the glutamate binds, this causes a movement of the lower part of the ligand binding domain that then closes up around the glutamate. And that pulls on these linkers here that are attached to the transmembrane regions. And when glutamate is bound and the ligand binding domain is closed, that causes the pore to open, and that's what causes the ions to pass through, the electrical current, which in terms of AMPA receptors is an excitatory current, and it makes neurons more likely to want to pass on a signal. So so this is how they work. This is how they open. They also have a separate process as well for when they close. So when glutamate is removed, then they can just let go of glutamate and close, so they go back to their resting state. If, however, glutamate levels hang around, either due to very high frequency or just some slow clearance of glutamate out of the synapse, they can stay bound to glutamate for a much longer time. And a separate process kicks in, almost a defense mechanism, to prevent excessive excitation, whereby the pairs of ligand binding domains up here will fall apart. And when they fall apart, even though the glutamate is still bound within the ligand binding domain, the tension on these linkers is lost, and then the pore will close off and terminate the electrical signal. Okay, so that's the mechanisms, or the broad mechanisms of AMPA receptor gating. So how do these PAMs work? How do these positive modulators of AMPA receptors work? Well, if we think about these receptors and as they desensitize, the PAMs work by binding at this interface between the two pairs of ligand binding domains. And so whereas in the absence of a PAM, this will fall apart and close the channel, in the presence of a PAM, the binding of the molecule kind of grips hold of either side of the ligand binding domain dimer and prevents it from falling apart. And so that boosts the signal. It slows down the termination of the signal. And so by this mechanism, the PAMs can lead to increased currents through the AMPA receptors. And that's what may be of benefit to patients with underactive receptors. Okay, but to understand the potential benefits, the potential for AMPA receptor PAMs to help in the case of loss of function variants, we need to understand more about these loss of function variants. So, as I've already shown, AMPA receptors have got a lot of moving parts and a lot of important sections within their protein, and so there's a lot of things that can potentially go wrong. So, for example, if we think about receptors that are underactive, there could be changes in the ligand binding domain, which means that glutamate will no longer bind to the receptor and therefore won't be able to produce opening. Alternatively, maybe glutamate can bind to the receptor, but there's some issue with the linkers or the gate that prevents the gate of the channel being open. So glutamate can bind, but the channel nonetheless won't open. Alternatively, maybe glutamate can bind, maybe the gate can open, but there's some issue with the ion channel, meaning that ions won't comfortably pass through the receptor, and again leading to limited currents. Going further, it's possible that there could be an unstable connection between pairs of ligand binding domains. So there is excessive desensitization. So desensitization is only supposed to kick in when the signal has been going on for too long. If it kicks in too soon, again, that would terminate the signal. And sorry, there's lots going on here. I'm sorry, it's the end of the day. This might push you over the edge towards madness, watching all this stuff happen. But there's one further option, which is the possibility that a variant could lead to an unfolded protein. So as proteins are built, they're built one amino acid at a time. And as those amino acids string together and you have a quarter and then half of a protein and so on, they naturally start to fold themselves into the final shape they need to make a functional receptor. And if a key position within that process is damaged, even though looking at it, it doesn't seem there should be any reason why it disrupts the gating behavior of the channel, nonetheless, it might mean that it prevents it forming its final shape. And so there are many different ways you can envisage variants might lead to GRIA loss of function. And there's no guarantee that any one of or all or none of these particular types of loss of function variant would be helped either at all or helped equally by an AMPA receptor PAM. So we want to try and understand as much as we can about all these potential options for GRIA loss of function, and then try and work out if there are any classes of these that are more or less likely to be helped by addition of an AMPA receptor PAM. Okay, so how do we go about doing our studies? Well, we're very much into the functional changes that happen. So that's we record the electrical properties of the receptors to try and work out in what's up with them, basically. So the way we do it is we use a cell line called HEK293 cells, and we do a transfection to them, which is a means of putting DNA that they don't normally contain inside the cell. And we can do this with GRIA DNA, and we can either put in a healthy copy of the gene, or we can put in a variant copy, i.e. one that's harboring a variant that's found in a patient. We then take a small patch of membrane off these cells once they've made our receptors, and then we activate these receptors with glutamate to try and understand how they behave. And that looks something a bit like this, whereby we position our patch of membrane on the end of an electrode in front of a glass tool that we very rapidly move from a control solution into a glutamate solution. And when we do that, we can kind of trick the receptors into thinking they're at a synapse, because normally at a synapse, there's no glutamate at all. Then all of a sudden, there's a lot very, very quickly. And you have to activate AMPA receptors in this way because they are so fast. If you try and do this to a whole cell, you will never get the rate of exchange required to understand. So that's why we choose this exact method. And when we do that, we can see you have a baseline, and then you very quickly activate your receptors. And then fairly quickly as well, they then desensitize. So the glutamate stays there, but then they turn themselves off. Okay, so what have we been finding? Well, this is a representation of a range of variants we've looked at, specifically in the GRIA2 gene. And so we have kinetic data. That's where we jump in and out of glutamate for 25 of these GRIA2 variants that I'm showing here. And the color coding is what you might expect based on what we've seen in terms of loss or gain of function. So the black dash line, that is the control That is what a healthy GluA2 receptor should do. It should activate very quickly, and then it should desensitize at about this kind of rate. And what we find when we look at all these different variants that have come up either in the literature, or we've had direct contact with clinicians, or we found these out through CureGRIN, is that these GRIA2 variants that are associated with disease produce a wide range of desensitization behavior. So we have 10 variants here, which have a slower desensitization. So this is a simulation based on our recorded data. And you can see in one case in particular, there's just no desensitization at all. And in some cases, the rate of desensitization is only slightly slowed. But of course, we need to do this for all variants. We can't only look at the loss of function variants. We don't know the loss of function until we've done this test. But clearly, for these ones in green, we know we can discount them now from our ongoing studies with AMPA receptor PAMs. There's no need to boost these receptors because they're already probably producing too much current already. However, for these ones here, these ones in red, they seem to have accelerated desensitization. So these are the variants that are likely to have their signal at the synapse is likely to be terminated too quick. And so these are the population we can look at. And so there are 6 of those. And also we have 4 variants here that produce no current at all. Okay, and so across these 25 variants, there's quite a nice symmetry to it. We have 10 variants with a slow desensitization, and then we have 10 variants combined with ones that have either fast desensitization or no current at all. So there seems to be a pretty even division between loss or gain of function. And then there are 5 here in the middle, which for whatever reason seem to be associated with disease, but we can't immediately see a problem like this. So we've already started isolating the particular variants we're most interested in looking at in terms of these PAMs. And, oh, sorry, that was a stray thing. However, cautionary tales, and this is something that I guess is probably seen more often with GRIN variants, where you can get variants with a gain of function type phenotype for one property, but a loss of function type phenotype for another. And so it's really important that we don't rush this. We make very, very certain about our loss and gain of function distinction before we head on. So for example, for these 10 variants with a slow desensitization, actually when we look more detail at the channel properties, they actually had a really low conductance, which meant the ions were passing through much less than they should. So that was likely to the loss of function due to a slow passing of ions or a low passing of ions was likely to be a bigger influence on overall behavior than the fact that it was a little bit slow. So one of those wasn't really a gain of function. Equally, for this fast desensitization ones, one of these actually appears to have an increased affinity, so it binds glutamate for much longer. So even though it desensitized quicker, nonetheless, it had a longer signal in some other measures. These neutral ones, one of these seemed to desensitize normally, but had a deactivation that was slower. So it, even though it desensitized at the same speed, if you just removed the glutamate, it would let go of it actually much more slowly. And then the real eye-opener for us was these couple that when we expressed them with a TARP gave no current. In actual fact, it was likely that these were very toxic for the cells, and all the cells that were expressing the protein we wanted to record actually died. And so we were left with the instant impression that there was no current, but in actual fact, these were enormous gain of function. So this ongoing work means that we've already helped to kind of work out the nuances of various of these different variants, but we are always very, very mindful that we have to get it just right. We can't start, there's no point wasting our time working on the wrong variants, and we certainly don't want to give the wrong diagnosis to a patient. Okay. Okay, and so just to finish with just one very brief result. I'm not going to talk about any of the actual PAMs we're really focusing on, but this was an effect of cyclothiazide, which is a classic PAM. It gives huge boost in response, but it's not really a drug. It's not really safe in patients. And when we look at over here, we have the wild type response, so the healthy glutamate receptor response here in black, and this is one of those fast desensitizing variants here in red. Now, when we throw on cyclothiazide, which is this super strong PAM, we show that we see this enormous current then appear, even out of the loss of function variant. Okay, and so the good from this just very preliminary experiment is it is possible to reverse this kind of loss of function with an AMPA receptor PAM. So even ones that are desensitized very, very quickly, it is possible to slow down their desensitization. But of course, the bad is that AMPA receptors with a complete lack of desensitization are not healthy. The way to treat a patient with GRIA loss of function is not to give them GRIA gain of function. Of course, what we're trying to do is to recover normal. So we're trying to perform rescue pharmacology to try and make their receptors look as much like a healthy receptor as we possibly can. Okay, and so as our project advances forward, we're going to keep on characterizing these variants as they come in. If you have a GRIA variant, or you know of a GRIA family who don't yet know if their variant is loss of gain of function, please do reach out to us and we can potentially try and help identify the nature of that variant. We want to investigate other PAMs, so not cyclophosphamide, but other ones that have a better chance of ultimately making it to the clinic, and they don't all have the same size effects. So some high impact ones can completely block desensitization, similar to cyclophosphamide. Now, this might mean there's a narrower safety window with those, because the danger is that you could go from a loss of function to a gain of function situation. There's also some lower impact ones that have a more limited effect, and these are the ones that have a greater normalizing potential for patients. And so we're certainly going to spend a lot of time looking at those. And all the time we're going to try and continue to innovate. We're going to look at our practices. We've got an awful lot of data from these fast applications to outside out patches, but we want to look at other ways of working as well, including computational approaches in terms of predicting the impact of these variants on potential synaptic responses. Okay, so just to finish then, so thanks very much to our collaborators. We've worked closely with Alan Bayet, who you heard from earlier, and Anders Christensen in Copenhagen, our colleagues at the UCL Institute of Neurology, and of course, the lab here at UCL featuring Ben Tag gets an asterisk because he's your guy. He's the one who will be carrying out the majority of these experiments for the next 18 months. Okay, thank you very much indeed.
Thank you so much, Dr. Coombs. Okay, so it looks like we do already have a question in the chat. If anybody has any questions for any of our speakers related to their projects, feel free to put them in the chat now. We're also going to shortly stay in this Zoom, but we're going to transition to Ask Me Anything next. So you can also feel free to add any other questions that you have in there. But we'll start with this first one that I see here for Dr. Coombs. How do dominant negative loss of function mutations respond to the sensitization assays? Are PAMs candidate therapeutics for dominant negative variants?
Yeah, that's an excellent question. I think I was probably going to say a line on that, but I skipped straight past it. You're absolutely right. If your variant leaves your receptor in a little puddle of protein parts on the floor, then there's not really much you can do with a PAM. However, there are a couple of things to say about that. First of all, it may be that you have a complete loss of function. It still looks like a receptor and it can be associated into a functional receptor. It still looks like a subunit, I should say, and it can form itself into an intact receptor, in which case, while our initial screen is to look at homomeric receptors, so all 4 subunits are the same, we also have assays where we look at the more natural form of them, which is a heteromer. So they have a couple of different subunits in there. So it's possible in that situation, you would have 2 dead subunits within a receptor, in which case the overall receptor would have a decreased function, but it would nonetheless be functional. And then you could boost the responses of that kind of half-active receptor. The other situation is when they really are just a completely unfolded protein, and that then gives you a variant which has the equivalent of what we call haploinsufficiency, which is where some patients only have 1 copy of a gene when they should have 2. And if you have a completely unfolded protein, you effectively have that same situation, in which case all of the receptors you have are healthy, it's just there's not enough of them. And because we know that PAMs can bind to healthy forms of the receptor, again, it gives the opportunity that might be something therapeutic. So it's not something we can look at directly in our experiments, but if we can identify that is the nature of the variant, it certainly doesn't prevent PAMs being a candidate therapy.
Thank you, Dr. Coombs. I don't see any more questions currently in the chat related to the talks. I did want to first take a moment to thank all of our families for raising funds for all of these exciting projects. All of your hard work and effort with fundraising and making donations has contributed to funding each of these. And as you saw, that there's a lot of exciting things going on and potential treatments, better understanding and characterizing GRIA disorders. So it's a really exciting time. And we thank you all for your hard work and for all of the researchers for their interest and for their efforts in order to bring treatments and cures for GRIA disorders. Oh, so it looks like we do have another question from Lana. Does this apply to all GRIA numbers?
Yes, it does. Yes, I should have made clear we focused on GRIA 2 because we first got interested when there was a big cohort of GRIA 2 patients published in 2019, and so we already had quite a few things to work with in that regard. But as Dr. Lal said, there's a lot of similarities between all the GRIA genes, and there's particularly in the most important regions, they're very, very well conserved. So if a drug binds to GRIA 2, it's going to bind to GRIA 1, GRIA 3, and GRIA 4. And so, yes, while our first work has been in GRIA 2, we also have loss of function variants in GRIA 4. Dr. Byatt mentioned about their paper that was just published in about the GRIA 3 gene, and there's a lot of loss of function data in there as well. So we kind of have a lot of potential material to work with. We are still very keen to try and make subdivisions in the different GRIA variants to try and work out exactly in what way the variants are loss of function. But yes, it basically, yeah, it applies to all genes, yes.